Good afternoon and welcome to NASA's Kennedy Space Center and the Forward Osmosis Bag Experiment Demonstration. Joining us today are Dr. Howard Levine, NASA Project Scientist, and Monica Soler, Project Engineer with Kinetic North America. We'll begin with some opening statements and then go into the demonstration.
So we'd like to show to you a tech demo project that's flying on the shuttle Atlantis this Friday. All right, it's what we call the forward osmosis bag. So think of it as a bag within a bag. And the outer bag you can introduce dirty water or water that cannot be drunk by the astronauts. On the interior, the inner clean bag, we introduce a high sugar solution concentrate. And just by osmosis, it cleans the water. It pulls the water through a semi-permeable membrane, selectively excludes elements, compounds, bacteria, viral particles that are not desirable, and you end up with a sports drink that maybe 4 to 6 hours later can be drunk by the crew members. This technology was developed at Ames Research Center, and it was licensed to a company, Hydration Corporation in Oregon. And they went through the SBIR, Small Business Innovative Research Program, to commercialize it. And they developed this product, which is for hikers, so that hikers out in the wild can put this into a stream, and water that may not be suitable for parasites, or suitable for parasites but not for drinking, can be cleaned in this way. There's also a version of this that has been modified for the Department of Defense, for soldiers in the field. The Coast Guard has also developed a version for salt water to make salt water drinkable. So the project will be flying on Friday. We're going to be, first we had to modify the bag, so this commercial product is not flight rated, as we say. It has materials that don't meet the stringent flight requirements of the International Space Station. So we created a flight version that has the proper materials. And Monica's going to go through a demonstration of how the crew members will load it up, both the dirty side and the clean side. And in the experiment, we're going to be flying 6 of these bags, 3 of which will be manually mixed by the crew, and 3 of which will not be. So we'll be seeing whether there's a requirement for them to be manually mixed or not.
Okay. So what we have here, what we'll be flying on Friday, we're actually going to be flying 7 of these forward osmosis kits. They're made out of Nomex, and inside of the kit it's going to hold a forward osmosis bag, an osmotic concentrate syringe, forward osmosis sampling syringe, and the input storage bag. And each kit's going to have a letter identifier, A, B, C, D, and so on, for the crew member to associate with. So as Howard was saying, we modified the commercially available off-the-shelf product and changed the materials from PVC to polyethylene for flammability purposes. And we also changed the ports to self-sealing quick disconnect ports for leakage purposes. You can imagine trying to pour in some water into one of these big red ports in microgravity. You'll have water flying everywhere. We also have your standard 60 milliliter syringe, which is going to hold fructose and glucose syrups and deionized water. As Howard had mentioned, it's going to be injected into the inner chamber of the forward osmosis bag. The sampling syringe is an identical version of the osmotic concentrate syringe, which will be pulling a 60 milliliter sample approximately 6 hours after each bag is induced. And the input storage bag holds 500 milliliters of potassium chloride and a methyl blue dye, which is your typical food coloring over-the-counter dye. And both of these items will be launched preloaded, and all of the lab work is being done at the Space Life Science Lab here at Kennedy Space Center. So to begin the initiation, the crew member is going to transfer all of the liquid from the input storage bag into the outer chamber of the forward osmosis bag using this forward osmosis pump syringe. So I'm going to rotate this to make it a little easier. So the crew member will ensure that the check valve that we have here is in the off position towards the forward osmosis bag. The Velcro that we have on the input storage bag is used to assist the crew member in pushing all the liquid towards the outlet of the input storage bag. So they can roll the bag just like that, and they're able to get all the liquid towards the outlet. And they'll be pulling liquid into the forward osmosis pump syringe just like this. And then once they have it approximately full, they're going to move the check valve in the off direction of the input storage bag and slowly inject the forward osmosis bag with the dirty water. Now this cycle should take up to about 8 cycles for the crew member to get as much water as they can out of the input storage bag. So I'll go through this quickly. So as the water lessens, they'll keep rolling the bag using the Velcro to pull the dirty solution. Now the purpose of the methyl blue dye is to wet the membrane. It's to dye the membrane. So when the forward osmosis bags are returned for a post-flight analysis, we're going to be able to dissect the forward osmosis bag and see how the liquid wetted the membrane, whether or not it was a uniform wetting or if it was splotchy. We'll do a couple more pulls. And then once they've gotten as much liquid out of the input storage bag as they possibly can, they'll use a dry wipe, which they already have up there, just to make sure that there's no spots that come out, to disconnect the QDs from the input storage bag and the forward osmosis bag. So as you can see, all the liquid is on the outer chamber of the forward osmosis bag. Then they're going to come and grab the osmotic concentrate syringe and inject the osmotic concentrate to the inner chamber of the forward osmosis bag. And the osmotic concentrate is the driver for the water to pass through the membrane. So the higher concentrated liquid pulls the potable water through the membrane. So once they're done with that, they'll disconnect. And as Howard said, 3 of the 6 bags are going to be hand manipulated. So hand shaken for approximately 2 hours. And this is just to compare whether or not mechanically inducing the bags will actually increase the flux rate of the water moving through the membrane, or if 2 minutes, approximately 2 minutes, yeah. So once they're complete with the initiation and the mixing of the bags, then they're going to restow them back into this forward osmosis kits and back into the storage mid-deck locker. And approximately 6 hours after this is done, they're going to come back and they're going to remove a 60 milliliter sample from the inner chamber of the bag using this forward osmosis sampling syringe. Very similar to the way we did with the osmotic concentrate syringe. They're just going to connect it and remove the 60 milliliter sample. I won't remove a sample right now just because there's only been a couple minutes since I've actually initiated the bag, so you probably won't see much of anything. And then they're going to restow everything back into the forward osmosis kit and back into the locker for return. All operations are going to occur within 2 days of landing, and all the post-flight analysis will be done here at the Space Life Science Lab at Kennedy Space Center. So.
Okay. And
So the key part of the science is 6 hours later, when the crew member goes and withdraws the 60 milliliter sample, you saw that this osmotic concentrate is yellow. Okay, it has a dye in it, a fluorescein dye. The sample that we collect will be diluted based on the water that's passed through the membrane. All right, so this is our microgravity sample. This is the most important sample of the whole experiment. And we'll be able to run this through a spectrophotometer post-flight and get an idea of the flux rate of the water passage through the semi-permeable membrane based on the dilution of this dye. This other blue dye, as Monica said, is to get an idea of the distribution of the water in microgravity on the semi-permeable membrane.
Okay, and now we'll take questions. Please state your name and affiliation and wait for the microphone.
Jim Siegel, Celebration Independent Newspaper. So let me get this straight. Nobody's going to drink anything of that's been sampled here or experimented with on this flight, correct?
That's correct.
And that, so could you describe a little bit about what this would replace if it was to be used on the Space Shuttle or the International Space Station in a little bit of detail, and how large would something like that have to be on the Space Shuttle or the Space Station in order to do that?
So we think of this primarily as an emergency contingency water system. So in the event that one of the normal physical chemical means of producing potable water went down, this could be used in an emergency situation. It could also be used in the future as part of an EVA, extravehicle activity suit. Some of you may be science fiction fans. You may have read the Dune series with the Fremen on Arrakis, and they had still suits. Well, this could be almost a first step towards recapturing the humidity from our sweat, from our breath, even from our urine, and recycling it and making it drinkable. So mostly, I guess, to answer your question, as an emergency situation, it's not meant to be a day-to-day water generation device. But it could also be used when the crew comes back on a capsule and they land in the ocean. It could be filled with seawater and potable, drinkable, a sports drink kind of product could be made that way as well.
And just to add to that, this is actually phase 1 of the forward osmosis bag. So we're using this technology demonstration to demonstrate the technology of the membrane in microgravity. So we've already been accepted as a SDTO or XDTO for FOB phase 2, so that we would be able to take more samples at an hourly interval and get a better curve of the flux rate as the water passes through the membrane. So this is pretty much just demonstrating the technology of the membrane for this particular flight.
And the key point there is that it's necessary to elevate the technology readiness level of this technology and demonstrate that it functions in a microgravity environment. And that's really the main purpose of what we're trying to do. And once we demonstrate that it does indeed function in microgravity as it does on Earth, then the program can take it and plan on using this technology in future endeavors.
Okay. Ken, you have a question?
Yes. Hi, Ken Kramer from Spaceflight Magazine. Describe a little bit, the shuttle astronauts will do this, not the ISS astronauts. And when you do bring it up as an emergency situation, yeah, that's what I was wondering. How many bags would you bring up, and has anything like this been flown before? Thank you.
Well, I'll take the first couple of questions. Yes, this is a sortie mission. So a sortie payload meaning that it will be launched and returned from on the shuttle. It will not cross over onto the space station. So we have one of the U.S. crew members, actually Rex Wellheim, will be conducting the experiment 2 days before they're returning. And